Today, we are diving deep into something absolutely incredible, the power that's locked inside our very own bodies. We're going to be talking about cells that hold the keys to growth, repair, and, well, maybe the entire future of medicine itself.
I mean, really think about it for a second. You get a paper cut, and a few days later, poof, new skin. You've grown, well, a lot since you were a baby. The common thread here is your body's amazing, built-in ability to just make more of itself. And no, it isn't magic. It's one of the most fundamental processes in biology. And that biological engine? It has a name: mitosis. This is the specific type of cell division that lets you grow, heal, and repair damage. Without mitosis, that little paper cut would just stay a paper cut forever.
So how does this incredible biological copy machine actually work? Well, first, a cell gets bigger, kind of bulking up its internal machinery. Then, and this is the most important part, it makes a perfect copy of its DNA, a complete second set of every single chromosome. Those two sets get pulled to opposite ends of the cell, and then it splits right down the middle, creating two brand new cells that are genetically identical. And that word, identical, it is everything. The entire point of mitosis is to make a perfect clone, right? I mean, you don't want stomach cells to start growing on your arm where you had a cut. You need more skin cells. Mitosis makes sure you get an exact copy.
Okay, and this is where we hit the central puzzle. If mitosis is basically just a biological photocopier churning out perfect duplicates, then how in the world do we end up with over 200 different kinds of cells in our bodies? From brain cells to bone cells to blood cells. Where does all that amazing variety come from?
The answer, it turns out, lies with a very, very special kind of cell. A master cell that can do way more than just make copies of itself.
So let's meet the stem cell. The key word you need to know here is undifferentiated. Think of it like a blank slate. It's a trainee cell that hasn't been given its job yet, and it has two superpowers. It can divide to make more blank slate stem cells, or it can differentiate, which is a fancy word for transform, into other, more specialized cells, like a muscle cell or a nerve cell. Now, not all stem cells are created equal. You've got embryonic stem cells, which are the most versatile of all. They can become almost any cell type in the entire human body. Then you have adult stem cells, like the ones in your bone marrow. They're a bit more limited, but still incredibly powerful, mostly creating things like blood cells. And hey, it's not just us animals. Plants have them too, in places called meristems, and they can grow into any part of the plant.
So we have these amazing biological blank slates, and their potential, well, it's opening up a completely revolutionary new chapter in medicine.
And when I say revolutionary, I mean it. The potential here is genuinely life-altering. Just imagine being able to treat paralysis by growing brand new healthy nerve cells to repair a spinal cord injury. Or for someone with diabetes, what if we could grow them new pancreatic cells that produce the insulin their body can't? The possibilities are absolutely breathtaking. This all sounds fantastic, right? A future where we can just repair our own bodies. But this leads us to the most complicated and, honestly, the most controversial part of this whole story. Where do we actually get these master cells? Especially the most powerful ones, the embryonic stem cells.
There are really two main paths that scientists are exploring. And you know what? Each one comes with its own incredible set of advantages and its own profound ethical questions. One method is called therapeutic cloning. Here's how it works. An embryo is created that is a perfect genetic clone of the patient. Then stem cells are taken from that embryo. Those cells can then be used to grow perfectly matched tissues or organs for that patient. And here is the absolute game changer. Because the DNA is a perfect match, the patient's body won't reject the new cells.
Okay, so let's put these two big ideas side by side. On the one hand, we can use leftover embryos from IVF fertility treatments that are donated with full consent. The big advantage here is that these embryos already exist, but the major downside is that the patient's immune system might see these new cells as foreign and reject them, kind of like a typical organ transplant. On the other hand, therapeutic cloning completely solves that rejection problem with a perfect genetic match. The disadvantage, you're creating an embryo specifically for the purpose of harvesting its cells, and for many people, that raises some huge ethical alarms.
And beyond just where they come from, there are other pretty big hurdles. There's always a risk of transferring some kind of infection when you move cells from a lab into a person. As we've seen, many people have very strong ethical or religious objections to using human embryos for any kind of research.
And then there's a massive biological risk. Stem cells are masters of dividing. If that division isn't perfectly controlled, well, what is cancer? It's uncontrolled cell growth. It's a risk that absolutely has to be managed. So while the human debate is this constant balancing act between promise and peril, the basic technology of cloning from master cells is already being used in a whole other kingdom of life. Let's take a quick look at plants.
It's pretty amazing, actually. Using the stem cells from a plant's meristem tissue, scientists can do some really cool things. They can clone rare plants to help save them from extinction, and on a massive scale, they can quickly produce huge numbers of crops that are identical and have really useful traits, like being resistant to a certain disease. That's a huge deal for farmers and for our food supply.
So, here we are, standing at a crossroads. We have this technology that could cure diseases that we've always thought were incurable. And at the same time, it forces us to ask some really deep, really difficult questions about the very limits of science.
Ultimately, for any of us to form a real opinion on this, it has to be about more than just the science. The key skill is being able to weigh all of it, the incredible medical benefits against the very real risks, and to seriously consider the tricky social and ethical issues that this powerful technology brings with it.
And that leaves all of us with a final massive question. This science is moving so fast, giving us abilities that honestly felt like science fiction just a few years ago. As we get this godlike power to repair and maybe even rebuild our own bodies, the biggest challenge might not be scientific at all. It might be a human one. Who gets to decide how far is too far?